Before you start, you should ensure that you have all of the components required for your PCR assay, as well as a clean and well-organised workstation. You will need to use appropriate personal protective equipment, including a lab coat and latex gloves. A clean workspace and appropriate personal protective equipment is as much to protect you from the reagents as it is to protect your assay from DNA contamination, either from yourself or the environment. Set up your workspace. If you are right-handed, you should have your pipettes on your right-hand side, with the tip boxes positioned nearby. Position the esky, which contains the reaction components, within easy reach of your left hand. Directly in front of you, you should have a tube rack, which contains a tube of sterile water, empty tubes to make up the bulk mix, and PCR tubes in a PCR tube rack. If you are left-handed, the above setup should be reversed. Before setting up a PCR, it is important to determine the number of samples that you wish to amplify. This should be noted in your lab book. Now it's time to set up your bulk mix. Before starting, ensure that each of your components is completely thawed. Vortex each tube and spin in the benchtop microfuge. This ensures that all of the liquid is in the bottom of the tube. You will set your bulk mix up in a clean, sterile Eppendorf tube. Label your tube clearly. Following your protocol, add each of the components, except for the Taq polymerase, to your bulk mix tube. Add the water last, as this is usually the largest volume. When adding the water, pipette up and down gently to mix your bulk mix. Vortex the bulk mix tube and spin the tube in the benchtop microcentrifuge. This ensures all of the liquid is in the bottom of the tube. Your demonstrator will add the Taq polymerase for you. Take your tube to the appropriate place in the laboratory and ask for the Taq polymerase to be added. Your bulk mix now has all of the required components in it. Now it's time to set up each reaction. Label your PCR tubes with your initials and a number. Dispense the required volume of bulk mix into each PCR tube. Add the DNA samples to each tube. Add water to the water control tube. Your samples will require vortexing to ensure that they are completely mixed. Vortex them gently and spin them in the benchtop microfuge. Submit your samples to the demonstrator who will run them on the PCR machine. Ensure that they are clearly labelled and follow any instructions given to you. Your demonstrator will load them into a PCR machine and run the thermocycle for you, and you will get your results next week.